everyone. Welcome to Biosite. In this lecture, we are going to discuss about tools of recombinant DNA technology. What is rDNA technology? So it is a laboratory technique of gene manipulation. So basically, we replace or we manipulate the genome of one organism with desired gene. So for this gene manipulation, we require certain tools in rDNA technology. Let's discuss them. So one is restriction enzyme, also known as restriction endonucleases, restriction enzymes or restriction endonucleases. And the second one will be the DNA polymerase enzyme, which is used to synthesize the new strands of DNA. And another one is ligase enzyme. So this ligase is basically for the joining of DNA strands and a cloning vector and a competent host. So these are the basic tools of rDNA technology. Let's go in detail. So restriction enzymes, the enzymes which are used for cutting off DNA at specific locations in rDNA technology are called restriction enzymes. So for their ability to cutting off DNA at specific locations, they are also known as molecular scissors. So as they are meant to cut the molecule like DNA, so they belong to a major class of enzymes known as nucleases. So there are two different types of nucleases. Two different types of nucleases are present. So one is hexonuclease, hexonucleases, and another one is endonucleases. So exonucleases are the type of restriction enzymes or molecular scissors which remove. So the first discovered restriction enzyme is HIND2. It was isolated from the Haemophilus influenza, a microorganism, by Smith, Wilcox, and Hilley in 1968. So today, more than 900 restriction enzymes have been isolated from 230 strains of bacteria. So these restriction enzymes are mostly from the prokaryotes. So the prokaryotes are the ones which are the sources of these restriction enzymes. So how do we name the restriction enzyme? So basically the first letter in the name of a restriction enzyme comes from the genus of the organism, the isolated organism, and the two second letters comes from the species of the prokaryotic cells from which they were isolated. For example, if you see EcoR1, this is the restriction enzyme used in rDNA technology. So how do we name this restriction enzyme as EcoR1, which means it is isolated from the Escherichia coli R gamma 13. This is one such strain. So the letter R is derived from the name of the strain, which is R. It's an R strain, and the Roman number 1 indicates the order of the enzyme from the bacterial strain. So this Roman number indicates the order of the strain, bacterial strain, and R stands from the name of the strain, whereas Roman number indicates the order and R indicates the name of the strain. So this is how we name the restriction enzymes. So what is the mechanism of action of restriction enzymes? So each restriction endonuclease specifically or recognize specific palindromic sequences in DNA. So here what are palindromic sequences? A sequence of 6 base pairs in the DNA that read in both strands in opposite directions is called palindromic sequences. So for example, so if you take for example, this is the a DNA sequence. So let me show you what or how a palindrome actually looks like. So if you take the sequences like this is the double stranded DNA sequence, which is 5 prime to 3 prime and 3 prime to 5 prime. So if you notice, let's take the base sequences, which are 6 base pairs: G, guanine, adenine, adenine, thymine, thymine, and cytosine. And the cytosine pairs with guanine, adenine, adenine, thymine, and thymine, and cytosine. So these are the palindromic sequences. If you can observe the sequence, which is from 5 prime to 3 prime direction, it stands GAATTC, in which 5 prime to 3 prime direction the sequence is same, and if you observe the 3 prime to 5 prime direction also, you can observe the both strands in opposite directions. The sequence of 6 base pairs in DNA that read in both strands. If you read the base pair sequence in the both strands in opposite direction, the sequence will be the same. So if you take 3 prime to 5 prime direction palindrome, so it also gives the same pattern. One is GAATTC. So in whatever the strand, whatever the direction, opposite direction you observe the sequences, they will be same. So this kind of sequences are known as palindromic sequences. So palindromic sequences. So the major function of the restriction enzyme is to recognize these palindromes or palindromic sequences in DNA, and it, after the recognition, it cut the DNA at the specific site. So if you take, this is the palindrome sequence. This is one sequence from the vector DNA and this is one sequence from the foreign DNA. So with the action of the restriction enzyme EcoR1, we cut the DNA at specific site. So the site is known as restriction site, where the restriction enzyme cut the sequences. So EcoR1 action upon its specific DNA sequence, the arrow indicates the specific site of the DNA and it creates the sticky ends like this. Sticky ends, and the sticky ends will be joined by the enzyme known as ligase. So the sticky end of this is the vector DNA and this is the foreign DNA. Foreign DNA and this is the vector DNA. So the sticky ends of both foreign and vector DNA will be joined or ligated by the enzyme ligase. So this is how we obtain recombinant DNA. So the some part of the DNA is from the vector and some part will be the foreign DNA, gives a recombinant DNA molecule. So the mode of action in which the restriction endonuclease cut two strands of the DNA at different locations So this kind of cut is known as a staggered cut, results in the formation of sticky ends. So when you observe this, so the eco-R1 cuts the DNA at different location. In case of vector and foreign DNA, gives rise to sticky ends. So here the cutting happens in case of vector DNA, which is plasmid, and the foreign DNA gives sticky ends like this, which you can understand the cut happens between two different locations, leads to successful ligation and the formation of rDNA like this. So this is all about the restriction endonuclease mode of action. And the second most tool is DNA polymerase. So DNA polymerase. So whatever the desired DNA fragments obtained, we need to amplify them for the efficient uses. So for that, we require an enzyme known as DNA polymerase. So the DNA polymerase enzyme should be in such a way that it must withstand the highest temperatures for its efficient action. So the DNA polymerase of the organism Dermis aquaticus bacterium is used in rDNA technology, and the Dermis aquaticus had DNA polymerase type tag polymerase. So tag stands for Dermis aquaticus. So the speciality of this tag polymerase is it can withstand the highest temperatures during amplification of DNA fragments. So what is the function of this DNA polymerase? It extends the primers, which are the chemically synthesized DNA fragments. So how do it extend? By adding the deoxyribonucleotides, results in the formation of a template DNA. And it shows repeated replication result in the synthesis of multiple copies of the desired DNA fragments, and this process is known as amplification. So how do the DNA polymerase contribute for amplification? Let's see the steps. So these are the basically the steps of PCR, which are polymerase chain reaction, in where we use the DNA polymerase so to amplify the desired DNA fragments. Firstly, we need to separate the double stranded DNA, and the step is known as denaturation. So at the temperatures of 90 to 95 degrees Celsius, we denatured the double stranded DNA, in which the parental strand, the strands of the DNA will get separated like this. So this is the first step, which is known as denaturation. And the second step, where you can observe the separation of the strands, and here comes the annealing, where you decrease the temperature to 55 to 70 degrees Celsius for the addition of primers. After that, after the addition of tag polymerase, followed by ending followed by the addition of tag polymerase and DNTPs, it efficiently synthesized the strands, DNA strands. So here the temperature will be raised to 72 degrees Celsius for the activation of the tag polymerase, where it can efficiently synthesize the DNA strand by using deoxyribonucleotides. So this step is known as extension, where finally we get the two amplified DNA molecules. So this cycle continues repeatedly to obtain the multiple copies of the DNA. So the ligase enzyme, this enzyme helps to join the sticky ends of the vector DNA with the desired DNA fragments, result in the rDNA, which we have already discussed in the mechanism of restriction enzyme action. And we have the cloning vectors in the rDNA, which are very, very important. So the cloning vectors are nothing but these are the carriers for the transferring the foreign DNA fragment into the suitable host is called vector. So vectors are used for the multiplication of foreign DNA sequences. So these are known as cloning vectors. So the commonly used cloning vectors are the plasmids. The plasmids are the most commonly used cloning vectors, and we have bacteriophages. There are some features for the vector. So there must be some properties for the molecule which we need to select them or categorize them as vectors. So the vectors needs to have a specific site for replication, and this site is known as origin of replication, and it requires a selectable marker for the identifying and eliminating the non-transformed cells and permitting the growth of the transformed cells. So after the take up of the desired DNA molecule, the cells are grown in a differential medium where the cells which obtain the rDNA will get separated from the non-transformable cells which do not get transformed at all. So for this identification, the selectable marker act as a tracking molecule, and the vector must have a single cloning site for the activation of restriction endonuclease, where the endonuclease cut at one specific site, and the vector should have the low molecular weight. So host is for the transformation with rDNA, is also required as a tool. So that kind of hosts are known as competent host, which are capable of transformation when they get rDNA. They get transformed. So this kind of host are known as competent host. So these are the tools of rDNA technology. I hope you understand this lecture. Thank you for watching.